very excited to present to you today, for the first time, primary findings from the AHHA trial. This trial was conducted in July to September 2019 as part of the Geonutrition project, funded by the Bill and Melinda Gates Foundation. A quick background to this trial. Micronutrient deficiencies or hidden hunger are widespread in Malawi. Felix Piri and colleagues recently reported a high prevalence of selenium deficiency in Malawi. This is due to soil factors. In large parts of the country, the soil is acidic and the native selenium is unavailable to crops. This leads to inadequate dietary intakes of selenium. This second map shows the likelihood that an adult woman in Malawi is selenium deficient, compared to the threshold for optimal GPX3 production, an important antioxidant enzyme. The red areas are deficiency hotspots. We conducted the AHHA trial here in Wimbi Traditional Authority, Kasungu district. Maize is the staple crop of Malawi, eaten daily by almost everyone. You can very effectively fortify maize by applying selenium fertilizers as the crop grows, a technique called agronomic biofortification. So the AHHA trial set out to test whether consuming maize enriched with selenium fertilizers can successfully improve selenium status in a typical rural context in Malawi. We conducted an individually randomized parallel group, two-arm, double-blind, placebo-controlled trial, working with adult women and school-aged children as participants. Ethical approval for the study was granted by the Malawi College of Medicine and the London School of Hygiene and Tropical Medicine. I'd like to flag Limbanazo Matandika's presentation on Tuesday of the ANH conference, where she talks about an innovative real-time ethics approach for ag nutrition trials. The protocol for the AHHA trial was published in the journal Trials. To prepare for this trial, we had to grow a lot of maize, some for the control group, where we followed standard agronomic practices, and some for the intervention group, where we followed standard practices and added selenium fertilizer. Here's an aerial photo of the maize under production at the Luana farm, under the management of Professor Patson Nalavata and Hastings Manesse. Only tiny amounts of selenium are required to agrofortify maize, equivalent to 1 to 2 teaspoons over a football field. We conducted formative fieldwork a year before recruitment began, and this informed our extensive community engagement program. This was essential for getting the participant communities on board with the trial. Please do check out Dr. Gabriela Chietsupiri's presentation on Wednesday of the ANH conference to hear more about this. This diagram gives you a quick overview of the trial design, from screening of households in the study area, through recruitment, randomization, baseline survey, the intervention, which lasted for 8 weeks, and the end-line survey. The primary outcome was concentration of selenium in blood serum at end-line. 96% of participants completed the trial, defined as giving blood samples at baseline and end-line. Blood samples were collected in mobile clinics by trained nurses into trace element-free vacuotainers. These were then spun down in the field, and aliquots of the serum were maintained in a cold chain back to the field base, then to Lilongwe, then under a material transfer agreement to the UK for analysis by ICPMS. We also collected socio-demographic data, measured participants' height and weight, and collected detailed dietary data for the adult women. The intervention involved distribution of maize flour, either control flour or flour enriched through agronomic biofortification with selenium. We provided enough flour for all members of participant households, since families tend to cook and eat their meals together. Bags of flour were labeled with unique QR codes so we could track their distribution. The trial was double-blind, and participants did not know which type of flour they received. Control equivalent flour was also provided to households in the study area who were not participating in the trial. This was done to reduce the chance of sharing flour between households and to minimize feelings of jealousy or stigma in the community. In total, over 200 tons of maize flour were distributed. So now to the results. You'll remember that the primary outcome was serum selenium concentration, and we worked with 2 demographic groups, women of reproductive age, and school-aged children. At baseline, serum selenium concentrations were similar between control and intervention groups. The majority of participants were deficient. Their serum selenium concentrations were below the threshold required for optimal production of the antioxidant GPX3. And here are the serum selenium concentrations at endline. So the intervention clearly worked. There was a substantial increase in serum selenium among women and children in those receiving the selenium-enriched maize flour, with the majority of participants lifted out of deficiency. There was no evidence of change in the control group. This is an exciting and important finding. It suggests that selenium deficiency could be greatly alleviated in Malawi by adding tiny amounts of selenium to commonly used fertilizer. The next steps are to work with agriculture and nutrition communities in Malawi to design and test programs and policies to roll out this intervention. So with that, I'd like to thank all my colleagues and collaborators involved in this work. Additional thanks to the Independent Trial Steering Committee and to our funders, the Bill and Melinda Gates Foundation. Particular thanks must go to collaborators from Loana, especially Dr. Alexander Kalambira and Leonard Banda. Also to collaborators from the Public Health Institute of Malawi and the College of Medicine. Thanks also to the fantastic team of research assistants, and of course to the trial participants and the communities in the study area for their cooperation and partnership.